Welcome to day 2 of the LabKey User Conference. Today, I have been asked to conduct. My name is Bernie Lee. I'm product manager for LabKey Biologics. And we have some really interesting presentations coming today. First, there's a product update on LabKey Biologics from me. We're going to follow that up with more time with me. I'm going to provide an electronic laboratory notebook preview from what we've been working on. And then I'll be introducing Kulas to talk through this story from their perspective. And we will round out the day by hearing from our very own Josh Eckels about some of the work related to Panorama and targeted mass spectrometry. So with that, I'd like to jump right into this product update. So many of you will not be familiar with LabKey Biologics, so I'd like to start today by giving an overview and update for LabKey Biologics. I've now been its product manager for about a year. And I only get more excited about the amazing advances and potential of this research space. Biologics research is very complex and in need of continuously improving tools to serve its variations. And we aim to do just that. So first, I'm going to give you a little bit of an elevator pitch, a little overview. And then I will walk through some key product highlights to give you a sense of what it's actually like to use the product. I'll describe our support and product approach. I'll get into our focus for this year, and then we'll talk a little bit about where we're headed in 2021. So LabKey Biologics is fundamentally a centralization tool for the many data types generated across the various teams that work to develop a biologic, and those can be many. It helps manage work requests, data capture, and soon we'll have an integrated electronic laboratory notebook, which all help to accelerate biologics development. And many companies, those different teams can very much become information silos, even though they're working on the same molecular entities, even though they're doing some related experiments, they hand off materials and data between each other, and they're working towards the same objectives. So LabKey Biologics provides a central source to register molecular entities, such as sequences, molecules themselves, and all the various expression system components. Now, all the stakeholders that are involved can know where to look for those data and relationships. Since the sequences and molecules can come from many sources, some internal, some external, we employ an automated classification and annotation engine to ensure consistent cataloging of those entities, duplication prevention, and calculation of some physical properties. LabKey Biologics also allows you to capture physical sample information. These samples can get manipulated over time and changed, derived, etc., as they're passed between scientists. So LabKey Biologics tracks those lineage relationships through generations to allow you to explore those results and to just explore that tree of relationships. At each of those different sample generations, data can be collected from various measurements. That assay data becomes more meaningful when integrated with known sample information. So LabKey Biologics provides structured assay data capture. Now, molecular entities obviously are not the only things that, the molecular entities themselves are obviously not the only things that affect your results. So we also allow you to capture information about your media recipe and the batch information associated with them. And lastly, all of these scientific operations have to be coordinated. So we have a work request system in place to facilitate some of that communication. So that's not exhaustive, but I just wanted to give you a highlight of what is here in the product. And then I'd like to talk a little bit about our approach, which I think distinguishes us somewhat. So the problems tackled by LabKey Biologics are common across very different organizations, from bioprocessing CROs and end-to-end R&D companies to those trying to create synthetic foods. And it has, of course, been particularly pleasing of late to work with some who are trying hard to reduce damage from our over-dispersed coronavirus. But that variety within such a complex process means the planning, rollout, and change management for software can sometimes be rough to navigate. So our approach is to provide a very close partnership with our support teams, technical teams, and product teams. We continuously share our expertise and what we have learned, working with others to help with training and data modeling, as well as troubleshooting as you go along. We also regularly seek your scenarios to better understand what each type of organization is doing and where the science is headed, and ask for product feedback so that our products and services can improve and grow with you. So all LabKey products are built to be empowering. What I mean when I say that is that when you're getting set up or wish to make a change, we do not want you to have to come to us and ask us to change the code and the software powering your LabKey instance. What we want is for you to be able to simply make the changes and understand the consequences with our support. As an example, you know which types of samples are prepared and run through your various instruments. So you can establish the connections and relationships between them. You may know what information about those samples and instruments you need to capture, but learn later that you want more, or you want to capture more information about a particular test or have a new test. And you can just make those updates in minutes. All right, so let me return for just 1 minute to my previous statement about seeking product feedback as part of our approach. That's not just a bromide. So I came from a biochemistry background and a human physiology research background, and I left the bench, frankly, because I thought the advancement of these fields could be accelerated more by having really good tools, having better tools, as opposed to having my hand in the experimental design itself. And that I'm pretty passionate about that. And that passion is for building and refining tools is easily matched by the LabKey company culture. Our account managers, software architects, dedicated user experience designers, and product managers have and will continue to spend countless hours discussing scientific operations and pain points with our users and prospects. That user feedback really is how we develop our products. And that has been a very heavy focus in 2020, with the express intent of making software adoption quicker by improving our user interface to be more intuitive. The changes we continue to make reduce hurdles for data management and reduce ramp-up time for new users. So work in biologics development, as I suppose everywhere else, is becoming higher throughput across the board, which speeds things up. But as you might imagine, this adds complexity to data management in some obvious ways. And we have moved to support its manifestations with our assay architecture. As I think, as Adam mentioned yesterday, that includes things like broader plate support for our assays. So we also know that some research questions can only be answered by combining data from many assays, or have data forms that require specialized views or visualizations to be interpreted appropriately. To those ends, we have empowered scientists to stitch that data together and extend those data views, customize those data views to meet their needs. And the last one I have up there is for ELN, but I'm going to continue to gloss over that for now, since the last half of my time this morning will be dedicated to it. All right, so where are we investing this coming year for LabKey Biologics? First, I want to continue our drive towards data while you work. A phrase I'm planning on trademarking because I think it's funny. You're also, of course, welcome to whistle while you work, but masks have seriously inhibited the whistlers among us. You're welcome to go try it. It's harder. Data while you work is, what I mean is removing barriers to scientists capturing their data while doing their experiments. It should be as easy as possible to take your data, put it in the right place so that you and others can easily make further use of it, including in other useful products, by the way. When you pull up LabKey Biologics, we want you to, and you want to quickly get right to where you need to do your work. That is why we're making more UX investment in streamlining navigation and improving our landing pages. We also plan on adding some nuance and power to the workflow management tooling that I mentioned in my overview, so that it can be better used to guide scientists in fulfilling experimental work and data gathering in line with related work. And again, completing the integration of our ELN so that all of the work that you have done to capture your data does not have to be repeated to get it into your documentation. Another focus in our product development will be accelerating implementation. So these may be seemingly small things, like creating a connection between your cell line samples and viability measurements for you, so that that's not something that you have to do. Or it might be connecting protein purification workflows and yield measurements, things that have natural pairings or natural groupings. And of course, as I said before, you're always empowered to make changes to these things. But the idea is to have some of this pre-configured to get you going faster. We all know that for good reason, many experiments follow very similar patterns, or frankly are redundant in many ways. And so another way that we'd like to accelerate adoption is by providing templates for various things. In this case, for workflows, common experiments, and processes. That should reduce how much setup needs to be done to make progress. In the same way that having notebook templates as part of this ELN, which I'll talk more about later, would reduce how many times you have to write up the same thing or copy paste when you're documenting. Given the inherent challenges of new software implementation, some of which I mentioned before with all of the change management, really every small efficiency gain adds up and really matters. So lastly, in terms of product direction, we have very regular conversations with groups doing all kinds of things like enzyme or strain engineering coming out of discovery. We talk to protein production CROs and those end-to-end biotherapeutic organizations. All of them demonstrate pain points in modeling the data capture and workflows for things like protein production, whether those are deep well plate operations or flasks or bioreactors or any of the myriad scales of those operations. And the same goes for a lot of the post-production processing, if you will, like purifications and various filtration methods that are used to get the material ready for analytical and preclinical and other work. It's again, we've heard that there are some pain points with the way that some current solutions, some existing solutions, whether it's a standalone piece of software or not, leave something to be desired in this space, and these operations are totally ubiquitous across the industry. So even as we round out our ELN development, we will begin focusing on bioprocess modeling. And because of our distinguishing approach, which I talked through earlier, I think that we're well suited to provide an empowering solution in this space. So I might have talked a little bit fast there, but that's what LabKey Biologics is. I hope that overview was helpful. I hope that brief review of what we've been doing was interesting, and now you have a clear sense of where we're headed this coming year. I want to give a special thank you to many of our good partners, in particular our Product Advisory Council. We have a number of people that are committed to talking to us about our product in a very constructive way, in a very candid way, and I'm very grateful for them and their time. We also have a really wonderful design, development, and test team, and I'd like to thank them as well. And of course, for our very supportive leadership that is helping me be a good product manager. So with that, I don't suppose anybody has any questions in mind.
I'll just open it up too and let people add questions in the Q&A section here. We've got a couple, Bernie, so far. So what are your main mechanisms for gathering user feedback for the product?
Sure. So main mechanisms. So the first one is well, I'd say most of them are in the form of conversation. So when we work with a company, a group, a lab, we have regular meetings with them to talk about priorities, progression, configuration, how operations are actually proceeding, and we use that time as well to ask and answer questions about product use, pain points, hiccups, those types of things. And so the dedicated account managers for each group, as well as me as the product manager, spend time processing that feedback internally, talking with our dev teams, and incorporating that feedback into our product strategy. We also, as we're doing development, we also go around soliciting specific user scenarios for the we'll call them features that we plan on developing, and making sure that our plans accommodate as many of those as is reasonable. And then we also perform user experience UX testing sessions once we've started the process. We have prototypes built and are partway through development. We spend time one-on-one with users, putting them through some of the scenarios and seeing how they interact with the page, and make adjustments to our plans of development based on what they do and what they say.
I'll just have the added plug too that you'll probably have in your next presentation that we are actually going to be emailing all the attendees if you're interested in providing user feedback for some of our features. That is one of the other mechanisms. I've got a couple other questions here, Bernie.
Yeah.
Biologics seems well suited for many scientific disciplines outside of biologics. Are there any plans to expand more generic data tracking into other industries?
It's an interesting question. So it might be illuminating to just talk briefly about how biologics came about. So LabKey server in general, which you heard Adam Rauch and others talk about yesterday, is our broad platform for doing exactly that. And it was there first. It's been there for what are we up to, 15 years, something like that. And LabKey Biologics is in many ways a specialized version of that for biologics development. So, you know, if the way that LabKey Biologics is set up is useful outside of some of the you know, the specific scenarios that I mentioned or some of the organizations that I mentioned, that's fine actually. So we love to have conversations about how one of our specialized products might fit with you, and if it doesn't, we have approaches that we can take using LabKey server to accommodate those. And you know, if we get, you know, sufficient and good feedback about another specialization that's worth making an investment in, we want to consider that as well. So the answer is that outside of biologics specifically, we usually begin by thinking about how the use of LabKey server, the platform in general, will be useful for that application. But we make it part of the conversation. So we'd love to talk to you about it.
And then one more question on from a community user of the licensing model or tiers that LabKey or LabKey Biologics has.
Sure. So I can give you a general overview. So we have different levels of premium edition for LabKey server, which we're happy to point you to that on our site to give you a little bit of an understanding of some of the distinctions. LabKey Biologics is its own tier, if you will. It's its own product. It comes with many of the features that LabKey server premium editions have. As Adam indicated yesterday, our products, LabKey Biologics included, is built on top of LabKey server, and so you can use and take advantage of LabKey server's power while simultaneously taking advantage of the specific specialization that LabKey Biologics is. LabKey Biologics has its own pricing model as well, and I'd be very happy to talk through those things another time.
And what about customization of the registry?
Sure. So this is in one of my early slides where I was talking about biologics overview. I mentioned that there is this molecular entity registry, and in this case, we're talking about various types of sequences, molecules, and expression system components, I think is what I said. So there's a lot of those that come pre-configured at product rollout, but you actually can also add your own, as in you can create entirely your own registry types, just as you can create your own sample types and your own assay designs, depending on which ones you have and what measurements you want to take. And you can create relationships between those entity types. Again, you don't have to ask us to do it, although of course we will provide you documentation and training and walk you through it if that's of value to you.
And you mentioned perhaps like a UX designer would you mind elaborating on the approach for a user experience that you're taking?
Sure. I can try. And if it's insufficient, I'm happy to work with our UX designer to come up with a deeper explanation of how that works. So when we do these UX testing sessions, you know, we really want, I don't know what's the right word to call it. It's almost blind feedback, as in we want to not necessarily give you a lot of information about what you are about to be interacting with up front. We want to put the software in its current form in front of you and give you a story, a narrative, a scenario to walk through, and watch you think about how you might do that, so that we can better fine-tune how intuitive you know, the navigation and interaction is with the software. Inevitably arises from that some questions or confusion, or sometimes you as the user think that there's you know, you doubt what you're supposed to be doing, but you actually click on exactly the right thing. Those are the types of things that we want to gauge. And as you go through and you ask questions, we may or may not be able to answer them directly because we don't want to give anything away. We want to make sure that we get your raw, unbiased intuition. So it's usually, I don't know what we do, 30 to 60 minutes, maybe. I think maybe for the sessions that we have planned, we're doing 45-minute sessions where you know, we'll give you a little intro, we'll give you a little scenario, and then it's pretty fun. It can be pretty interesting. We want really candid feedback. We want you to think out loud as you go. It's kind of fun to just take a look at something that you're unfamiliar with and just talk about it as you play with it. And it's really useful for us. Sorry, I hope that's more in depth. I hope that's helpful.
I'll just jump in here real quick. My name's Matt Yakubowski. I'm user experience designer here at LabKey. Just to round out some of what Bernie is speaking to here, user experience process is really part of our entire development process. It's not a single stage of what we do. What he is alluding to is some user testing sessions that we will occasionally do. It may happen at different phases in our development and design process. But the ultimate goal is really ensuring that the software that we are making is designed to actually suit the goals that you want to accomplish using the software. It's not strictly about navigation per se, or a particular thing on the screen. We mostly want to be sure that we're incorporating all of the needs that you have, all of the pain points that you might have with your current software or solutions, and really bring that into a more well-rounded experience within our software, so that ultimately we're helping you work smarter, not harder. And that's really kind of our goal when we have integrated this UX process into our agile development.
Awesome. Thank you, Matt. Thanks, Bernie. I think we're out of time, and so the remaining questions that folks have answered, we will we'll send you an email as a follow-up, but also feel free to submit a question on labkey.com, and one of us will get back to you as well. Thanks, Bernie.
Thank you, Hannah. And thank you, Matt. Seriously. Swooping in to save me. Okay. So let's move on. I finally get to stop deferring on this. So as I hope I've described, LabKey Biologics can provide that valuable structured data centralization. But as we have worked with closely with the many users in our community, we heard that significant work is still often done by researchers to properly document cross sections of their data. That is that there's one or several ways the researchers store and analyze data, and then a separate method for notebook keeping. And that can lead to all sorts of problems, or at least headaches, like data duplication between different sources, poor coordination with others, and lots of wasted time, none of which is particularly good. So with that perspective, it will come as no surprise to you that our in-development biologics ELN has direct access to the data that you are storing in LabKey Biologics. It provides the ability to reference samples, assays, any of those registry entities, those molecular entities, files, other experiment data, all in one collaborative notebook that secures your IP. And unlike paper notebooks, obviously, and many available ELNs, our connection to your data really helps you maintain that single source of truth, prevents that duplication and extra work, and is backed by the power of LabKey, which you saw yesterday and which I mentioned just a few minutes ago. So there's a heavy collaborative nature in this, some of which I'll be able to show you and some of which I'll simply have to describe for now. But that includes co-authoring, on-page discussions through commenting, and the ability to reference data that another person has prepared. It's not something you don't have to do all of it, in fact. And all combined, these can mean big efficiency gains and, as I said before, headache reduction. So you can imagine for yourself how much time you or your scientists spend stitching data together for your documentation. If it's even 10% of your time, that could be up to like a couple hundred hours per year per researcher. And our plan is to really help scientists spend less time digging for data to document and more time on the satisfying stuff, like learning something or advancing a particular cause or treatment for a disease. Okay, so with that, I'm going to lay out a little demo scenario here so we can all get on the right headspace, and then I'm gonna try to walk through that scenario a little bit so that you can get a good feel for what this is going to be like. So we are a biopharma company. We have a couple of biologics in the pipeline, including work on a therapeutic antibody for COVID-19. Now, of course, all of our candidate therapeutics are important, but there is a little bit of time pressure on this one, as you might imagine. And last week as a scientist, I started an experiment that I know needs my attention. We were running a capto affinity purification and some size exclusion chromatography, going into the weekend, and I was told that this was done and the data imported by my co-author. So first thing I want to do is go and get that documented quickly, and then see if anything else needs my attention. So I'm going to jump in and show you around with the big disclaimer that what you see is not final. This is a preview. Many of our planned features are still in development, some of which I will talk about later, but they're still in development and can't be shown yet. And the ones that you see are subject to change. So. All right, so I arrive at my notebooks page this morning, and I see my 5 most recently accessed notebooks. And being presented with those really helps me maintain continuity in my work. And sure enough, here is this capto affinity notebook follow-up that I started to construct for our molecule designated PY43. Started by me and my co-author, Steve. By the way, this is the same Steve that is doing our awesome fundamentals training sessions each afternoon. He also happens to be an excellent scientist. And I can see the rest of the things that I accessed and their state and their authors. And once I open up this notebook, I can double check that my entries are constructed as I remember, so that I should be able to just add the things that I know are missing, and move on and submit it for review. So So first I can see some of the overview details, titles, dates, times, project designation, some of the actions I can take. I can get an overview of some of what I have going on in this notebook. I can see my various sections. In this case, I have a very, very reductionist purpose for this experiment, where I have made a reference here. This is a color-coded reference to the previous purification that was done on this molecule. I can see it was also done by me and Steve. It was a cation exchange chromatography setup. And if I wanted to, I could even click through and explore that notebook for reference. But I don't want to, since I just did that last week and I don't need to be reminded of what it is. I can also see in my methods section that I constructed this table that has some information about my various mobile phases and quantities and any notes on what's actually being performed. I can see the materials that were involved here. That includes some information about my column and my buffers, as well as these color-coded references to the samples that were, in this case, pooled and put into this, put through this purification run. You may have noticed, but I will point out that those 5 samples referenced here also show up in this overview pane. In fact, all of my references show up in this overview pane, so that I have an easy way of checking to make sure that what I expect to have here is here. So coming back down to the rest of my remaining sections, I see some of my results here. I see a basic table that I put in here with some summary information, as well as some basic titer results. But what I don't see that I'm expecting to see, or I'm sorry, what I don't see that I was not expecting to see that I was expecting to have to add are the my SEC run data, as well as my the trace for this purification and the SEC instrument setup. So I want to get those added. As I emphasized before, this notebook, all of the notebooks, have direct access to the data captured in LabKey Biologics. So knowing that that SEC run has been imported somewhere, connecting it to my notebook is really easy. In this case, well, actually in all cases, all I have to do is type / and it opens my reference menu. So this is where I can see, where I can look through and find any notebooks that I want to make reference to, any experiment groupings that I have configured, all of the different samples that I work with and have registered in the system, the various assays that are available to me, and any of the registered entities that I might want to access. All color-coded to help you distinguish them. In this case, as I said before, I'm looking for my SEC assay. When I go into assays, I can see all of the existing assays, assay designs in use in the system. As I mentioned before, all of these are configurable. Whatever you actually need in here is what will be in here. I'm looking for a particular SEC run, and we present you with all of the most recent runs that have been imported listed here. Now, let's just, I happen to want this PY43 molecule, the one that was run over the weekend, but let's just assume that I didn't see it there for some reason. It's easy enough for me to simply type in what I'm looking for. In this case, I know it's PY43, and I know it's a BEH run, and I can simply select that, and we convert it into this assay run. I can see in the tooltip summary information about that particular assay, and if I wanted to, I could even click through to this and examine the results. So we got that added, which is nice. As I said before, I also wanted to add my trace and my SEC instrument prep. So in this case, my instrument prep is just a file, which I happen to have in a different screen, and I'm just going to drag over, and you'll notice that I can drop that file wherever it's appropriate. I'm gonna put it in my methods section, and it's just going to attach it. I can see that it's a Word document. I can download that and review it again if I should so desire. I also want to paste my trace in here. I have that image from somewhere else. I have that image from the run, and in this case, I'm just going to copy and paste that in. And since attaching the file and making reference to my run, those things are reflected in this overview pane. Here I can see the same information for this particular run, and I can see my file that's associated. So now that everything appears to be in place, I think I'm ready to submit, except that I remember that we have an established desired order for my entries for this type of an experiment. And it's not what I have here, so I'm going to quickly reorder my entries. We actually want materials first, and we want our results last. We actually want to look at the trace before we look at our results, because we're weird. But voila, they are reordered, and I think we are ready to go. So with that, I can submit this notebook for review. We again get summary information where if I so desired, I could reorder these sections again. I can make sure that I have the right file here, and I can go and sign this. Signing is as easy as, and I have this pre-configured, but signing is as easy as selecting a reviewer or multiple reviewers, providing a deadline should you so desire. In this case, I'd like a quick turnaround on this. Any comments or specific things that you want to look for, and then it's as simple as re-authenticating. Hopefully I can type my password right the first time. Excellent. So my reviewers have been notified that, that this has been submitted and is waiting for them. And you can see on this card that might have been submitted. Okay, so my first priority is out of the way of getting that particular set of experiments completed and documented. And now I want to make sure that no other COVID work is blocked by me. So we're back on this page. You can see my recently accessed, but I'm going to come down to my main notebook listing. And I can see that there is a notebook awaiting my review. So I can click on that, and I see that it is a COVID notebook that my coworker Angie has submitted to me. You can see that it's locked once it's submitted. So let's do this next. So I can come in here, and it should look very familiar. It's reviewed in exactly the same format that it's constructed. To keep expectations the same. I can begin my review, and I can look for the things that I expect. I can examine what has been written. I can click through and examine these particular molecules or vectors or the other information that's referenced or written up. I can look at the plots that are put in here, and I can examine, in this case, the protein production worksheet that led to this, should I so desire. Now, if this matches my expectations, I can sign it, which looks exactly like the signing when you're the author. Or if, in this case, I happen to think that, hey, maybe there's some QC information that I don't see, I can send this back to Angie and say, please add QC information and send it back to her. And what you'll find is that we track within the notebook the back and forth between author and reviewer with the comments there. And so now Angie will get will be informed that there's something else that she needs to go in here and add, and she'll have the ability to unlock this notebook and add the information that I've requested. Okay, so now that that's all taken care of, and just to be conscientious, I am going to come down here and filter on my projects. You'll see I have a couple of them built out here. Just make sure that there isn't any other COVID work that someone forgot to assign or that I've otherwise missed. And I can see that everything of mine is in a submitted state or approved or is being worked on elsewhere. So I can be sure that I've taken care of everything that I wanted. Okay, so that's what we're ready to show so far. We are targeting the end of the first, we're targeting the end of H1 2021 for initial release of this in LabKey Biologics. So keep an eye out for more news on that as the time approaches. There are, however, a number of other high value features planned that are worth mentioning that I couldn't show you because they don't exist yet. The first one is commenting. This is really a key contributor to the collaborative nature of this ELN, along with what I said before, the co-authoring, ability to reference data created by others. So when writing your notebooks or reviewing, you'll be able to comment and discuss particular entries within the notebook. And this should make for a very rich and easy way to give feedback. I showed you some of the filtering that's helpful for finding what you need, but a search function will also be available for particular contents or authors. You can imagine the utility. If you are at a decent sized company, you may have hundreds or more of particular notebooks, but want to find just a few that use a particular instrument, for instance, or you know, a particular instrument and a particular person. And that's what the search will be there for. You may have also noticed I did not mention any kind of exports from the ELN, but there will be two to start with. One is standard PDF generated from what you wrote, pasted in and referenced. The other one is in some ways more useful for handoff and collaboration. We work with people that receive big PDFs from, you know, whomever else they're working with, but actually they need to make use of the data that's in there, and so they begin the manual process of extracting all of the information that's in that PDF, which of course is incredibly painful, time-consuming, and potentially error-prone. And so we plan this directory export that will have all of your referenced data in there as tabular files, which can simply be extracted and made use of. I mentioned in the Biologics product direction the efficiency value of templates, and hopefully that's made a little more real to you with the walkthrough that I just gave. If you're going to perform, you know, a dozen stability tests a week or a dozen purifications a week, we don't want you to have to do a ton of copy pasting and reconstructing all of your different entries. So we plan to provide the ability to just create a notebook from a template. So you can just walk up and basically fill in the components that are missing or different. Very similar to what I did in the demo, frankly. And then, you know, submit and move on. And the last plan work that I'll mention is some higher order composition, if you will. So notebooks are fine-grained, and they tend to contribute information to an overarching set of questions. You know, my one purification isn't the end-all be-all of this particular biologic development. It's part of you know, an overarching set of questions. So commonly, you know, groups of notebooks have critical conclusions, you know, extracted from them and put into scientific reports. for rapid consumption and decision making. And we plan on adding a scientific report tier on top of the existing notebooks with connections to the underlying notebooks that allow you to do that composition here as well. And much more. So as I've said several times now, our design and development involve gathering significant intelligence through the conversations and those UX sessions that that people have asked about. And we are, as you may have seen in the conference emails that went out, we are ready to broaden participation in those sessions. So have a look at those emails for some additional information. And if you would be interested, and we'd be very grateful if you would, interested in contributing to our product quality, please have a look and sign up. So I have to basically thank most of the same people. In this case, you know, unbounded gratitude to our good partners and product advisory council group. We have a fun name for our EL engineering team, for that development team. So thank you for your creativity, persistence, and good teaching of teaching me about what you're doing, and again, supportive leadership. So I guess we don't have quite as much time to run through questions this time, but I'm very interested if any of you have any questions about this ELN highlight.
Yeah, Bernie, great job. There are a few sort of like questions, so I'm going to try and bundle them as much as possible, but of course we'll get back to everyone individually to help make sure that these questions have been answered. So one of the questions is really around sort of the customization of the ELN, and you know, how easy is it to customize? Is it customizable?
Excuse me. Yeah, so I assume you're talking about the, you know, a particular notebook. And so assuming that that's true, it is entirely customizable, as in, I guess I could show you this. Maybe it's not wise to go further into a demo that I didn't necessarily plan on doing, but I think I should just be able to jump all the way through this. So by default, these are actually empty, and all of the entries that I had pre-constructed in there are completely customizable. Their name, their content, their order, their format, start date, project association, title, all of that stuff is completely controllable by you, which again is empowering, but on the flip side, that's why we want templates as well. So you have the ability to make changes to these things, construct them from scratch, configure them entirely on your own, but also when you want to just power through from existing formats, you have the ability to do so.
And another sort of grouped question from several folks is whether or not you can register samples or entities or add data sort of directly in the ELN, or if those need to be registered first.
Sure. So a little bit of nuance there. So you, as you saw in one of my notebook entries, I had a table in there. I had various other pieces of data that were associated with a particular experiment. You can put sample information, ad hoc tables, whatever you want in there. Right now, the things that you put directly in the notebook that aren't already imported or registered elsewhere don't maintain that same connection. So as an example, if you are but you can still put them in there. So if there are parts of LabKey Biologics that you don't use for some reason, broadly, but you still want to capture certain information about your samples or something in the notebook, there's no problem with doing that. It's just that if LabKey Biologics is your data centralization tool, you have that power, you have that connection.
And then sort of another question I'll paraphrase that have come from a variety of folks too is where ELN is available, if it's just available in biologics or other LabKey products.
Sure. So the answer is first, LabKey Biologics. We're going to finish this for LabKey Biologics, and then based on demand, user feedback, and our planning, we certainly want to consider making it available elsewhere. It's of course a little bit different to have to have it for something as customizable as LabKey Server. So don't expect that to immediately follow right after release of this for LabKey Biologics, but it's Biologics first, and then we definitely want to consider expanding it for use in our other platforms.
And right now, I think that wraps up most of those questions.
I'm gonna end on time. That's so great. Okay, thank you so much, Hannah. Okay, with that then, I would like to introduce our other speaker today. We are about to hear from Tara Kulas from Just Evotec Biologics, who's gonna kind of tell you this story from a very different perspective. Tara is a scientist in the high throughput analytical sciences group at Just Evotec, and also manages the company's current ELN. She and her team are working closely with us to develop what I just showed you. Please welcome Tara.
All right, let me take over the screen share. All right, can everybody see my screen and hear me?
Yes.
Great. Okay, hi everyone. Today I'm going to talk about the quest for an ELN we actually want to use. And I need to click my slides. All right, so I work at a company called Just Evotec Biologics. We're located in South Lake Union here in Seattle. And as our name suggests, we are a biologic company. And this is a picture of our small family. I put small in air quotes because back when this photo was taken in 2019, there was about 90 of us. And since then, we've almost doubled in size. But we still have that small company feel. So it's still a small company in my mind. This is me. I work in the high throughput analytical sciences group, as Bernie mentioned. And I also happen to be the ELN administrator here. So who are we? Well, we're an integrated design company. But what does that really mean? Our company is focused on technologies, and we're focused on developing a technological platform that will help accelerate the development of biotherapeutics and substantially reduce their cost and development time. So our mission is to design and apply these innovative technologies to help dramatically expand global access to biotherapeutics. And we do this by being a super cross-functional company. And our goal as a cross-functional company is to knock down those silos that you tend to see at big biotherapeutic companies. And that will help us enable data transfer amongst those groups and also enable our technology. And at the center of our company is our in-house integrated technology platform called JDesign. And the core of JDesign is a common data management system, namely LabKey, that helps centralize and integrate our data coming from the four main groups at our company. Those being discovery, molecular design, process design, and manufacturing design. In discovery, we leverage machine learning to create large, diverse, and manufacturable discovery libraries. In molecular design, we leverage our in-house suite of proprietary computational tools that help enable prediction of the best molecules and conditions for development. Over in process design, we're really focused on optimizing the process. So that means cell lines, vectors, media, chromatography, filtration, biophysical and formulation development tools, and high-resolution analytical capabilities. And then in manufacturing design, we're focusing on disposable technologies and intensified processes to help create a flexible and relatively low-cost biomanufacturing facility. But what's key to note here is our solutions are data-driven, and that flow of data is huge between our groups. So that's what's really important to our success here. And so I'm going to start by talking about our workflows at Just and how those relate to LabKey, where all of our data lives. So I'll start with our upstream workflows, talking about JDiscovery, cell line development, and media. And at the center of all these workflows is LabKey Biologics. This is Brendan over in JDiscovery. Most of his day consists of creating nucleotide sequences, constructs, and samples, and putting all that information into LabKey. Over in cell line development, we have Pauline. In her pre-COVID spare time, she likes to spend time posing for pictures behind blank walls with antibodies. And most of her days consist of managing her transfections, selections, and productions. And she leverages project workflows and LabKey's API abilities to help create cell passaging, production, and cloning automation workflows, which help increase her capacity. So previously she was able to do 4 stable pools, and now with this API automation workflow ability, she can go up to doing 24 stable pools. She does a lot of inventory management as well, keeping track of all those cells that live in her freezer. Sample generation and lineage is really huge for this group. So keeping track of all of those cell lines, constructs, and intended molecules that are coming from that JDiscovery group. And assay submissions. Of course, everybody wants product quality data, so that assay submissions get submitted through LabKey and head over to us in the analytical group eventually. Over in media development, we have Abby, and most of her day is spent creating and tracking media batches. And so you can imagine with creating media, there's quite a bit of raw materials, ingredients, and mixture management, and inventory management to keep track of everything that she needs. She also does quite a bit of sample generation and assay submission for product quality as well. And all the media that she's making gets sent back to that cell line development group and off to the bioreactor group. And keeping track of all this media data in LabKey is what allows us to then leverage tracking how media affects our cell line and how media affects our bioreactors. Continuing with our workflows, I'm going to move a little bit further downstream. And again, at the center of all these workflows is LabKey Biologics. Over in the bioreactor group, we have Abigail, and most of her day consists of tracking her bioreactor runs. She does quite a bit of sample generation, and she leverages LabKey's API abilities again to create run sheet plots to track every single day of her bioreactor runs. She gets all of that media from the media group, like I was saying previously, and with that media comes all the data associated with that media. And this allows her to look for media effects in her bioreactor runs, which is super cool. She does quite a bit of assay submission for product quality as well. And so that will get sent over to the assay group and the analytical group, which is me later. Over in purification, we have Jan. All the material that he's purifying comes over from the bioreactor group with all that data associated with it. When he has that material, he's able to create material requests, and again, has a bunch of high throughput purification workflows that he's created that allow him to go from previously purifying 8 samples a day to maybe 296 well plates a day. So you can imagine producing that many samples also means he has quite a bit of bulk assay requests to do. And all these assay requests come to me over in Analytical. I also get some material to be purified from the bioreactors and do a little bit more quick and dirty purifications to create material requests and assay submissions and send all of that assay submission data right back to myself where I run those assays. After running those assays, I get a lot of raw data, and most of my raw data analysis happens in Empower, and we've created LabKey APIs as well to upload all of this Empower metadata right back into LabKey. Over in the drug product design group, we have Mark. Mark deals with hundreds of samples a day too for his formulation and biophysical studies. And so he uses sample generation as well and uses the experiment tool in LabKey to help track all these hundreds of samples. He does quite a bit of assay submission as well, and then also uploads all of his biophysical assay data into LabKey. So all this data is living in LabKey, but where does it go? So it goes off to our data science and molecular design group, and they are ultimately what's tying everything together here at JUS. This is them. This is our COVID stitched together photos, since we haven't been able to get them together since COVID times. But these guys are what is fueling our company. They can often be found sitting in front of their dual monitor setup, looking at antibodies and digging through tons of data sets to find trends and hits. They get all of this data that's coming from all these groups I just discussed via LabKey Biologics. And all of this data is then allowing them to do deep data dives and push out valuable analytics and learnings that feed right back into all of our groups to improve our processes, help improve optimization, and effectively is what enabling us to increase our capacity, increase our speed, increase our quality, and lower our cost. So this is everything that's fueling our company. Oh, and then there's the long lost ELN that's lingering in the back of everyone's mind months down the road. And So why are we forgetting about our notebooks? Like, what's the hang up? Well, the reason our documentation is often getting dropped is mainly because there just isn't a great ELN solution out there, and it's not integrated into any of our workflows. And this is really common in most industries. Most people are tending to do their ELNs after the fact because it's not integrated in their workflows. They keep all of their data somewhere else, and then the ELN is kind of an afterthought. So I just kind of want to talk about what our current ELN looks like. I'm not allowed to mention its name, but it'll help you understand kind of why ELN is getting dropped. So our current ELN is a paper and glass solution, but almost too literally. So assessing a bunch of criteria, including the UI, the ability to attach files, document upload size limits, functionality, and cost, it really just doesn't perform well. Although it is supposedly compatible with Excel, Word, and PowerPoint, we're often running into issues where we can't actually upload these files and they fail to upload consistently. Our document display is limiting us to both size and view, so uploading those documents is capped. And unfortunately, when we attach files, those files aren't actually visible during the sign-off phase, so most of our sign-offs are happening blindly, which is not the point of an ELN. So after being crowned the ELN administrator and being fed up with our ELN for too long, I decided to form a team of scientists from each area of research, namely the ones I just described, to try and find us a better ELN. So we decided to start by evaluating 3 out-of-the-box ELN options. My lawyer insisted that I was not allowed to name these options, so for the purpose of this talk, I will be referring to them as solution 1, 2, and 3. And we assessed a bunch of criteria, including UI, features, ability to do file attachments, ease of sign-off, data accessibility, and cost. Solution 1 is a task-based ELN, and unfortunately it had quite a cumbersome UI that required you to create tasks for each step of your notebook. So when you wanted an introduction, you were stuck making a task for that introduction. Materials and methods, another task. And unfortunately, this just got really endlessly long. And when you went to go sign off all of that data, you also had to sign off each individual task. So although the data, the ELN was cheap, it really didn't make us want to write our notebooks more. And so we all agreed this wasn't a good option for us. Solution 2 is ELN that's designed for small molecules. And so this could definitely be tailored to be a biotherapeutic ELN as well. But what we found was, again, problems with the UI. The notebooking window was really small. It required you to do quite a bit of scrolling when you had large file attachments. And everything was displayed in line. So you can imagine that when you're displaying Excel files that have hundreds of columns or multiple tabs, having all of that data trimmed is essentially cutting out all of the data. So again, just not a good option for us. Solution number 3 is kind of the gold standard ELN out there. It's a task-based ELN as well. And despite being the gold standard, it was just not as clean and simple as we wanted it to be. It had too much fluff and also excessive features that we already had in LabKey, namely sample registration and inventory. And in our minds, why duplicate that? If we already use a system that we love, why would we want that in another system? And at the end of the day, it was extremely not cost effective. And so with our growth here at Just, none of these options were good for us. But the biggest thing was the data accessibility. At the end of the day, all 3 ELNs still had the data living outside the ELN. So we would still be an afterthought as an ELN. It wouldn't be part of our workflow, which is what we needed. So we decided to go back to square one to define what the ideal ELN looked like for us. We want something with a simple and clean UI. So I think everybody can agree that in order to have a notebook that gets used, you need it to be simple to use and easy to adopt. We want something with easy and powerful searchability. So similar to LabKey, where we can dig up hundreds of samples, dig up assay requests from years ago, and have all of that data really easily accessible. We want the same thing in our ELN. So we can dig up the data that lives in our ELN, namely our explanations of all of our data. We want that to be easy to find. Obviously, we want the ability to view attachments at each ELN phase. This is the biggest hang up in our current ELN. Not being able to view those attachments during the sign-off is super awful. So wanting to be able to view those attachments while you're making the notebooks, while you're reviewing the notebooks, and after you've reviewed the notebooks. One of the things that we saw in a lot of ELN options out there is this ability to reference. Namely, in those ELN options, that's referencing users and referencing other notebooks. But we wanted to take this one step further and also be able to reference our data. So for us, that means referencing samples, assays, and experiments in LabKey. Obviously, a functional and valid sign-off phase is very key to an ELN. You can't have an ELN that is purposeful without this, and our current one, having to blindly sign off data, is not that. Something with collaborative notebooks would be great because as a company that is super cross-functional and collaborative, we're often collaborating on experiments, and why duplicate that data in 2 ELNs when we could just have one ELN and work together to create that ELN. And we want something that is a notebook and just a notebook. Like I said, we use LabKey for everything else in our workflows. We don't need to duplicate that within our ELN. And most importantly, we want an ELN that lives with our data. And so this is kind of where the light bulb went off, and we realized, why don't we ask LabKey to build us an ELN? And they agreed to it, and over the past 6 months or so, we've been working on this ELN with LabKey, and they've been helping us make this a reality. And so now that you've seen the demo, I kind of just want to talk about how this translates back to us and our workflows here at Just. So the simple and clean UI. Like Bernie showed, the homepage is exactly what you want it to be. It has quick access links to everything you need, allowing you to easily toggle between all the different experiments you have going on, easily search for data that exists in other notebooks, and access those tasks that you need to do. So check to see if you have notebooks that are waiting on you, ones that are overdue, ones that have been returned to you, and ones that you've submitted for review. So having this clean, simple UI is going to make this really easy to use, really easy to adopt, and help keep up with our notebooks. Again, simple and clean UI in the notebook actual writing phase, it's exactly what you want it to be and exactly what you expect. Something that is super customizable and something that has, simple, plain text fields, but also the ability to add in attachments in line and display those images that you would like. The ability to reference is probably the single most biggest thing in simplifying our workflows, as it's cutting out that time sink of duplication for us. That's a huge time sink for us. And Bernie mentioned this earlier, but it's so true. Unfortunately, right now, the way we do ELNs, the reason we're doing them months later is because nobody wants to have to sit there and just copy and paste data left and right when they already have that data existing somewhere. So simplifying that workflow and having all of that data easily linked in is what is huge to making that time sink disappear. And so being able to link in samples, assays, experiments, other notebooks, and then things like mixtures and other registry items is going to be great. And having that be able to be linked in right away. So previously, where you'd want to give sample lineage details or assay data details, you'd have to copy and paste that into your ELN. And now we can simply just reference that sample and have a direct link to its lineage, or reference that assay and have a direct link to all that assay data. And then thirdly, the ability to sign off data with ease. So the data sign-off phase often is something that is slowing us down because people tend to forget about signing off notebooks. And if there are problems with some of these notebooks, you're stuck going to find them and telling them, hey, can you fix this in your notebook so I can return it to you, and blah, blah, blah. It just takes forever. So having something that's easy to submit for review, easy to review and give feedback, and then have it be done is going to definitely enable quicker turnarounds for our notebook reviews. And so from that homepage, I'm ready to submit for review, and right at the top, there's a button that brings me to the next page. It shows me this great summary of having all of my entries, all of my attachments right there. I can go to the signature page, request who I want it to review by, give them a deadline, and it's done. And then on the other side, the reviewer sees exactly what I saw. So they see my notebook exactly as it exists with all of those referenced items, all of those attachments that are viewable. And it really makes it easy for them to just go ahead, go back to that signature page, give comments, put their username and password, and be done. And the biggest thing here, I think, will be in the future, we'll have the ability to comment directly in the notebook. And that's really going to enable our collaboration. And I think collaboration is huge at Just, and with many other companies, that's something that's really lacking. And being able to collaborate on those notebooks and allow for that, quick turnaround is really going to take out some of the time sink. And so our path forward, despite this being a demo product, is super bright. right now our ELN and our data will be living in the same place. And so everything that's coming out of LabKey is now just going to feed right back into it, because that's exactly where all of our data is living. And so the biggest thing here is choosing to work with LabKey is probably the best company we could have chosen to partner with us for, because they're constantly working to help us grow and continue improving and enabling us to make those improvements. So while this is currently a demo product, this is just the beginning. And as we continue to demo this product and as we continue to use it, we're going to find out that maybe we need to make some changes here and there, and how maybe we can improve our workflows. And LabKey is there to help us do that. So now our ELN is no longer going to be an afterthought, but rather it's going to exist where our data already exists. And with that, I want to just thank LabKey for helping make this ELN a reality. Thank you guys for listening to my presentation. And thanks to LabKey for giving me an opportunity to present on a product that I'm super stoked about.
Thank you, Tara. Thank you so much. So if anybody has any questions, I would greatly appreciate you adding them to the Q&A for Tara. I do already have some in here, so perhaps while I am asking Tara, other people could paste theirs in there. So this one seems fun. How have your workflows as well as your ELN needs changed, if at all, during COVID times?
That's good. So given the fact that we are, cutting our time on site, most of our time on site now is just purely being in the lab, and the interactions that we've had with people have obviously cut down. Being a company that is like super cross-functional and works in open space, now we don't really have those conversations that were so easy to have. Everything is happening online. And so with our workflows, that obviously is slowing things down, because we're not being able to just easily walk up to somebody's desk and be like, hey, can you get me this data? It's now it's requires, chains of email threads or Hangouts messages. But I think in terms of our ELN, this is, if anything, like showing us more and more that we need a collaborative ELN space. And so I think once we have that, ability to collaborate on notebooks and ability to comment on those notebooks, both during the actual notebooking phase and review phase, which is something that's later to come, it's really going to help us accelerate that whole ELN process. And our workflow is then going to have that ELN in place with it, which I think is what is really our hang up right now, is that our ELN lives so far out of our workflows, and our workflows for us are super efficient and work really well for us. And we're constantly trying to improve and optimize them and, add more workflows. So bringing that ELN in there is just going to help make everything work more smoothly.
Thank you. I've got another one on here that you should find interesting. Let's see, how do I summarize this? Okay, so it sounds like you'll be switching over from an existing one. Can you talk a little bit about how you, like what the rollout plan looks like for making a switch over, or for rolling out an ELN from a paper-based system looks like at all?
Yeah, so our current ELN, we had to renew our ELN just to, keep consistency with our ELN as we demo this product. And so the team of scientists that I put together is kind of our LabKey user group, and we will be the ones beta testing the notebook to kind of help the rollout phase. So once we have that first version, we'll be beta testing that, and it'll give us an opportunity to find and look for improvements that need to be made and see how it fits into our workflows and kind of what we can make better and how we can improve on it. And then once we've hit that stage, we'll bring it out to the rest of our scientists. And because it's already existed in somebody's hands in every single group, it'll be really easy for there to be kind of a point person to help teach the ELNs. I'm sure I'll give a presentation to demo the ELN and teach people how to use it. And so for me, it's pretty smooth. Our data transfer is going to be pretty easy in the sense that we're kind of just going to export everything that exists in our ELN and archive it for the time being. I don't think it's necessarily worth it to us to have all of that data exist in LabKey right now. So that's not happening for us, but I think that the transition will be really smooth. All of us are super familiar using LabKey, and so we kind of understand how the workflows work there. And I think it's going to be super easy and quick to learn how to use this referencing ability and making notebooks super easy.
Thank you. There's a couple more in here. So is your vision that your coworkers will fill out their ELNs in real time, kind of as they collect and import the data? What do you expect that to look like?
I think that's the ideal vision for any company, is that you have a notebook that you're doing as you do experiments. Obviously, that becomes hard if you don't have computers in every single one of your lab spaces and you have small lab spaces. I think that kind of exists for us, but I think the goal is that because it's going to be in LabKey where all of our workflows exist, and so we're all heavily using LabKey for just our day-to-day as is, it's going to be really easy to maintain those notebooks as you are real-time doing those experiments. And because you won't have to worry about data duplication, again, it's going to be really easy to be like, oh, those assay results got uploaded. Let me go back to my ELN and just tag those, because it's super quick. That's a minute of your time instead of maybe 30 minutes where you have to take all that data, compile it into whatever view you want it, make sure it uploads correctly, make sure it displays correctly. And that is no longer going to be something that people will do. So I see it functioning as more of a real-time ELNing situation. I know that many people will not use it as such, because the truth of the matter is most of us do our experiments in the lab and try to get out our data as quickly as possible. And then we remember, oh yeah, we have to ELN it. So I think it's going to allow for both, but hopefully it will transition us to, instead of doing our ELNs months down the road, maybe it'll be like a week down the road.
Very good. And there's another one on here about some of your workflows, not specific to any particular workflow, but in general. It's asked, how do the FDA's quality by design guidelines interface with your workflows?
I don't know if I can answer this question. The FDA quality by design guidelines.
Yeah, so I think it's okay not to necessarily answer directly about the FDA's guidelines, but maybe you could talk about how you have designed your workflows to meet with the both scientific and regulatory criteria related to the biotherapeutic development that you do.
Yeah, so I will say that all of our manufacturing side of things does not live within LabKey, just because all of the GMP regulations there are pretty tight and strict. And so we have a different system for all of that work. But in terms of, keeping up with being like ready to send all of our data out as necessary, that's where the ELN comes into a big play. So like if we're working on a product that we know needs to go to an IND soon, those ELNs need to be done in order for us to have valid data places. And so I think having our workflows as they exist now, they really enable all of the speed and the capacity to go through our lab really quickly, but the ELN not being part of that really slows down that phase of having all of those documents ready for a regulatory review. And so I think that having all of it in LabKey now or the ELN included, is really gonna help keep those two together. And so it'll help us prepare quicker, because right now we're probably tailing behind, and it's usually like, oh, I did all the work, but I still have to ELN it, and somebody's bugging me about getting my ELN done so that all those files can be reviewed. I don't know if that really answered it.
That's okay. We can follow up with this on that particular question another time. Thank you, Tara. That is all of the questions that are in the chat right now. So thank you so much.
All right. Thank you.
Okay. With that, we can move on to our last speaker for today.
And Bernie, before you hand off, if I could suggest, I see a number of questions about kind of ELN rollout, timing, compatibility outside biologics, et cetera. I know you touched on that before, but if you just want to kind of recap, because it seemed like there's excitement, which is great, on how people get their hands on it. Just kind of recap and invite people to those usability sessions.
Sure, happy to recap. So, LabKey Biologic, I'm sorry, the ELN at first will only be available to people using LabKey Biologics. We hope that we can work with our our partners and design teams to also make it useful in our other products, that being LabKey Sample Manager, which you will hear lots about tomorrow from Hannah, and hopefully as well in LabKey Server itself. The particular rollout timing of any of those things I'm sorry, the rollout for anything other than LabKey Biologics is unknown. There's a lot of planning and discussion to be had about those. I also see that there are questions about some of the capabilities within the ELN, such as cutting and pasted images directly. Yes, you can definitely do that kind of thing. I don't know if you recall during the demo, I added a trace for the purification that was done. That was an image that I simply had on a doc off screen, copy, paste straight in. So if you have anything like that you want included in line in your text, no problem, that works straight away. Let's see. Looks like most of the rest of these are questions about how it works, how it might work elsewhere. So maybe that is sufficient for now. As Josh said, a reminder, we would appreciate a diverse input on product quality, product utility, functionality. We have a set of the UX testing sessions that both I and Matt so kindly joined to talk about. We have some of those planned, and there is a sign up in the conference emails that go out for some of those sessions. So if you have any interest in seeing more, contributing to our product quality, and we would love to have you sign up for those and join us. So with that, I think it is time to hand off to our very own Josh Eckels. Since you are on, I will go ahead and let you introduce yourself, Josh.
Thanks, Bernie. So my name is, as Bernie said, Josh Eckels. I'm VP Engineering here at LabKey. I head up our San Diego office. And I'm here to talk about Panorama and LabKey support for targeted mass spectrometry. So, one of the disadvantages, of course, of being virtual this year is that I don't get to see your faces and get some sense of what's making sense and where I'm just talking crazy. Please go ahead and keep the questions coming. I'll try and keep an eye on the chat window and maybe be able to answer them as I go. Otherwise, we'll certainly loop back on them at the end of the session. And with that, kind of do a little intro to mass spectrometry for those who aren't familiar. I guess the plus side to being virtual this year is that for people like Tara, who know what HPLC stands for, I won't get to see your grimaces as I muddle through kind of this very basic layperson's introduction. But basically, a mass spectrometer is a fancy instrument that measures mass to charge ratio of ions. So what you do is you take something that you want to analyze with some set of molecules, you feed it into the instrument, it gets an electrical charge, and then it goes into magnetic field, and based on how much it deflects, you can measure this mass to charge ratio. So I kind of think of this as taking like a ping pong ball or a golf ball and throwing it into a cross breeze, and you get a very different deflection. For the ping pong ball, it's going to get deflected right away, as opposed to a golf ball that's going to go much farther before it kind of hits a wall or hits a fence or something like that. And so this can be done for both what's referred to as large molecules, which is basically proteins or peptides from a digested protein, or what's generally referred to as small molecules, which is basically everything else. So these can be drugs or lipids or metabolites or basically anything that's not a protein. And then what the instrument does is it's basically kind of counting how many of these things it's detecting in a given time slice. And then you can take all those time slices and do all sorts of amazing analysis on it. And so here's basically one of these time slices. So you imagine this is a bunch of ping pong balls going down, and then this is all of the golf balls that are a little bit heavier, for example, in my crude analogy. And then targeted mass spec in particular, as you saw, that's part of the title of this talk, is kind of a specific usage or specific mode of using one of these mass spectrometers. And so really what you're doing is you're targeting the expected set of ions or molecules that you're expecting to be there in your experiment. And so this is different, kind of a contrast to what's often called discovery proteomics or mass spec, where you're kind of doing a more open-ended, saying, oh, whatever comes through, I'll try and analyze it. This is, you're telling the instrument, hey, look at about this time for something in this mass to charge ratio range and really focus on it so that I get much more quantitative data of that, as opposed to trying to go broad and potentially missing parts of the data that you're interested in. So there's a bunch of different variations of this. There's a lot of different instrument types as well, kind of different variations of a mass spectrometer that have pluses and minuses for different usage modes. But I won't go into details, but basically kind of started out as, oh, I want to target one thing at a time, or I want to target multiple things at a time, and kind of building out these workflows and the capabilities of this type of analysis so that you can now measure thousands of different analytes that you're targeting as part of one of these experiments. Okay, so Tara, everyone else, you can unplug your ears now, and I'll get on to the specifics of what we're doing. So Panorama is really tied to a great Windows-based desktop analysis program called Skyline. So this is developed by the McCoss Lab at the University of Washington. His primary developer is Brendan McLean, who actually used to work at LabKey, and so it's great to keep collaborating with him and the rest of his team. And this is a project that started a little over 10 years ago, and it's got a huge user community, a lot of people doing great things and continuing to push it to work with new instruments and new types of new modes of analyses. And one of the things that I think is really distinguishing this from other targeted mass spec tools is that it works across all of the different mass spec vendor file formats natively. So there's no conversion step like you might have to do with many other tools. And so the Skyline team works very closely with the mass spec vendors to make sure that it's compatible with the new instruments or the new modes of usage. Ion mobility, for example, is something that they added in the last, say, 4 or 5 years, based on those capabilities coming online for the instrumentation and demand from their users. And Skyline really started with a proteomics focus, but they did a massive amount of work to have great support for small molecules as well. So if you've been holding off because you're doing small molecule, but you didn't think it was compatible, it's been there for quite a while, and I encourage you to take a look. So now let's talk about the LabKey side. Panorama is the module within LabKey server that really knows how to import that Skyline data. And it's very closely tied to Skyline. I've had people ask before, oh, I'm using MaxQuant or some other targeted mass spec analysis software. Can I import that into Panorama? And really the answer is not directly. Panorama knows Skyline's file formats, and we work with them to keep compatible with new versions of Skyline. So we're always kind of playing leapfrog or catch up to make sure we're current. And there are some great workflows to go directly from Skyline into Panorama. And I'll talk about those a little bit. But the McCoss Lab have been co-developers. Vidisha, for example, Sharma from the lab has done a lot of the Panorama development. And we've been at that since about 2012. And really the reason it kind of lagged the initial version of our initial release of Skyline is because people needed to accumulate enough data, enough Skyline files to have a reason to start putting them in some sort of data repository. So that's kind of when Skyline came along, is when people got tired of having a file share or emailing these Skyline files around, especially as they got bigger and bigger as these analyses grew. Panorama kind of came to help manage all that curation and searching and collaboration. And we've got a bunch of kind of different usage modes within Panorama that I'll get to in a moment. So the overall workflow is that we've got our mass spec acquiring data files, and it spits out a raw file of some sort. Again, like I mentioned, it kind of depends on the specific mass spec you're using and the vendor of that instrument, what that looks like. But Skyline takes that in directly, and then you curate a Skyline document. And you're saying, basically, here are the peptides or here are the molecules that I'm expecting to be present in this data. And then here are the transitions, basically how they kind of break up inside the instrument, so that you can further validate that, yes, that is the molecule that you're interested in. And you take that kind of Skyline template, think of it as, here are the 500 peptides I expect might be there from these 50 proteins, and or the 500 small molecules, the lipids that I'm looking at, for example. And analyze my raw file, see what's in there, and then import those results into Panorama. So that goes into the LabKey relational database behind the scenes. You get the web interface over that. You can do all the great API access and SQL queries and reporting that you're used to from other LabKey capabilities and modules. And then you can push that back into Skyline so you can download that original file again to refine it. Let's say there's a problem with it where you don't like exactly what section kind of the chromatogram that Skyline selected to say, here's the area under the curve kind of between these two boundaries. I want to go and adjust that, open it up in Skyline, and update that, push it back to Panorama. We'll keep track of the versions. We'll let you, kind of see that history. And it's really kind of this iterative workflow. And then you see over here, we've kind of got an express lane bypassing the Skyline user interface. And so this is really designed for QC, your system suitability. I'll talk about that in a minute. But it's a great way to be able to automate these workflows. So there's a whole command line interface to Skyline. So if you don't want to open it and see your data, you just want to have that analyzed by script, you can put together a pipeline quite simply where one of the end steps of that analysis is to load it into Panorama. So within Panorama, we've got kind of 4 primary usage modes. And so the way we've done these is as separate folder types within LabKey. So you'll create a new folder. You'll say, this is Panorama. I want to be doing targeted mass spec in this folder. And then I'll prompt you, okay, which of these is the best fit for what you're doing in this folder? And so I'll go into more detail on each of these in a couple minutes. But really what we're trying to do is have a user interface that directs you to the types of reports that you're most interested in, that kind of helps enforce expectations, that if you're expecting that you're going to have the same type of data over and over, that it says, hey, you tried to upload a file that doesn't match that pattern. So that's probably an error. And otherwise, just helps smooth that pathway so that you don't have to do a bunch of setup each time you create a new folder. It's kind of pre-configured and ready to go. So the first and kind of where we started really is what we call an experimental data folder. And so this is really kind of just like the repository mode of using Panorama. So you're generating all these Skyline files. Maybe they're related in that you're updating a given document and adding new raw files to it, or saying, oh, let me kind of curate exactly what peptides are the best representations of this protein, and I want to focus my analysis down to these, instead of looking at every single peptide that might have been coming from that protein. And so you upload your data. You can do that either by dragging your file into LabKey and clicking import, or the Skyline team added some options directly in Skyline where there's a menu option where you say upload to Panorama. And you do that by telling it, here's my Panorama server. So that could be your in-house server. It could be a LabKey hosted server, or it could be Panorama Web, which is a server that the McCoss Lab hosts for the community, and you can get a project there. And you give it your username and password, just like you would if you're doing any other kind of external API access to LabKey. And then you can upload your data directly from Skyline, kind of eliminating that copy and import step that you might otherwise have to do. So a lot of the views, if you're a Skyline user, will be pretty familiar. We're not trying to replicate every Skyline view in Panorama or vice versa, but a lot of the commonly used ones we recognize have a lot of value in both places. And you can do searches, so you can say, look across all my data and what was all the files, all the analyses where I looked at this protein or peptide or molecule. And just like any other LabKey folder structure, you can organize your data by study or project or instrument or the person who's conducting it, and then be able to do queries and views across those folders to kind of see that aggregate set. Version tracking is really something that wasn't part of our initial development, but we saw there's a huge amount of value in giving people ability to keep track of those versions. So this is a common question that we get asked for LabKey generally, which is, what's your audit history? What do you keep track of? If I upload a new version of this file, does it replace the other one completely? Does it kind of link with the other one? And so here we've done quite a bit of work to make sure we keep that full history. So if you upload 10 versions of a given file, that we're automatically connecting those and saying, aha, it's not just that we've got a list of 10 files and you can sort by the date that you imported it. Really, we're automatically linking those together because Skyline is helping us keep track that they're the same document with a unique ID for each one. And then you can go ahead and in Panorama, see how many versions of this file do I have? And then what are those versions? And click to see what was the initial one, what was the the second one, what was the most current one, as well as just saying, in general, only show me the most recent one. I want to have those others for archive purpose, but I don't need to have them cluttering up my list most of the time. And so one of the initial use cases we had for this is something called a method build. So this is, let's say you've got 10 proteins that you're looking to do some quantitative analysis on. And you're trying to figure out what are the 3 best peptides to help do that analysis for each protein. And so you might start out by saying, okay, let's look at all the peptides that are part of those proteins. Okay, now let's accumulate some more data. Okay, these peptides tend to be well behaved in the instrument. They don't degrade very quickly. They give me nice consistent results. They correlate with each other, or if they don't, that's a sign that something's gone wrong. And so I want to narrow that down from my initial set of 10 peptides to 5, and then my final 3. But I want to keep track of all my incremental steps along the way. And then more recently, instead of just tracking version 1, version 2, version 3 of the Skyline file, the Skyline team added some really robust audit logging on the Skyline side so that every edit that you make to that Skyline document gets logged. And so that might be, I change some quantification settings. So I change like a mass tolerance or something like that. Or I deleted a peptide or a transition, or I added some to that document. And so really this is fine-grained detail on exactly who and when a change was made to that file. Skyline's keeping track of all that, embedding it in the file as it goes. And then when Panorama imports that, it displays all of it. It connects that with the separate versions of the file that are separate copies that we're importing as well. And gives you that full history. And so really, if you're using Skyline, I encourage you to make sure that feature is turned on. For various reasons, it may not be. in older documents, if you've had ones that you're working with for a long time, there's a menu option to enable audit logging going forward. And really, this is something that we're looking to connect with some of the compliance features in the enterprise version of LabKey Server to satisfy regulatory needs. So to establish that, yes, we had this data on this date, and here's the manipulation that happened to get to this point. So think CFR Part 11 or similar types of compliance and regulatory needs. So we've got a little more build out to do there to make Panorama kind of more the repository of record for that. But we've got all the pieces and definitely interested in feedback from folks who have those needs, and especially if you're doing targeted mass spec in particular. Another set of views and reporting that we do in these experimental data folders is for quantitation. So the primary way that Skyline implements this is with a calibration curve. And so essentially what you do is you have a series of samples where you spike in your analyte at a known concentration, and you analyze those to see how much signal you get. Basically, what is the area under the curve for the observation from the instrument, and then create a calibration curve for that data and plot your unknowns against that to figure out what their concentration was. And so these are all set up in the Skyline document, and then Panorama automatically picks up on those and will calculate, like Skyline does, the concentrations for your unknowns, and then gives you nice interactive plots, as shown in the screenshot here, and then also calculates things like figures of merit. So basically to say, for my controls, this is not unknowns, but just kind of validating that against my curve, I get something within a percent CV, for example, of the expectation. How is my instrumentation? How is my assay performing? And so you get these nice summary views to really help you do monitoring of that. And then similarly, we've been doing work for pharmacokinetics, PK calculations. We have on our list, but we haven't yet implemented pharmacodynamics, PD. And so these are all things that we've heard from groups. Hey, we're doing this with this external tool right now. How can we help fold that directly into our Skyline and Panorama workflows? Quality control folders is something that we're really excited about, and we keep on getting lots of great feedback on. Hey, can we help monitor this? Can we monitor that? Can you give us this type of analysis? This is kind of really built on some capabilities that we did for some other assay types earlier in LabKey for Luminex, and we've generalized this. It's a premium feature to be able to create these types of plots over any data. But the great thing about using Panorama is it knows these types of metrics are relevant for you. Things like retention time or peak area, peak width, et cetera. And so this is where that auto QC kind of bypass on that earlier diagram comes in, is that you can set up a Skyline document to say, here's my normal mix of peptides or small molecules that are kind of representative of the types of data I want to be doing for my primary analysis. And I want to run those, say the first run of the instrument of the day, or every 10th run, or whatever the cadence is. And then set up a Skyline document to say, here are the things I'm looking to monitor. Use this auto QC utility where basically you point it to say, here's where the template document is. Here's where the instrument's going to be writing new files when it does one of these QCs or system suitability checks. And then auto QC will see that happen, analyze that file without needing to open Skyline at all. Behind the scenes, update that in Panorama, and then give you these great plots where you can say, okay, for this peptide, for retention time, basically when did it show up in my 30-minute mass spec run. I expect that to be consistent. I expect it to be about 15 minutes in. And really, if everything's working absolutely rock solid and consistent, this would be a straight line here at, say, 15 minutes. But you can see something went wrong. It shifted by 2 minutes. Or here, something happened. It became significantly more variable, as well as shifting to something closer to the 14-minute mark. And so this is really a great way to monitor the instrumentation to make sure it's behaving as you expect, and therefore you can trust all of the experimental data that you're generating on it. And we've got a lot of kind of early adoption. This tends to be a great entry point for people into Skyline and Panorama, because you don't even need to be using Skyline or Panorama for your primary analysis. You can say, right, I've got to be doing system suitability. I've got to be doing these checks and validation. Let me set that up, let it run in the background, see this data. We've got premium features where it'll email you if something's, if you've got a certain number of outliers, so things outside your normal range, like this point right here. And you can also curate with some premium features exactly what metrics you want to be monitoring. So maybe for you, in your setup, the peak width isn't that helpful. You get a lot of false positives. It's okay. You're not worried about that. You just want to focus on retention time and peak area. So you can curate that list of all the built-in metrics or analyze your own custom ones via a little setup and writing a query to let Panorama know exactly what you're looking to monitor. That's not one of those built-in ones. So this is a screenshot of AutoQC where you're pointing it at your Skyline template document. You're telling it where to look for these new files to be acquired from the instrument, some various settings. And then on the Panorama settings tab, you tell it, and here's where to import it into Panorama. And typically we have one Panorama folder per instrument. You might slice it a little more finely to say, oh, right, for this type of analysis on this instrument, track it there, that type, track it there. And then everything that runs through the instrument and gets deposited into this folder just automatically shows up in Panorama going forward. We've got some nice dashboard views to help summarize this. So if we've got, say, 4 different instruments that are all being monitored, we get the latest information. Is AutoQC actively running on that instrument, even if it hasn't uploaded anything recently? And again, this respects all of our customizations to say, here are the metrics I want to be looking at. Let me click through, I've got a lot of outliers in this one. Let me focus on what's going on, get that instrumentation back into alignment, or update the Panorama to say, no, this is the new expected behavior. Maybe I changed something like swapped out the column, and I've got a new expected retention time for these things. So I retrain Panorama to say, here's the new normal range, use that going forward. Multi-attribute method support. So this is a great connection with some of the biologics scenarios that Bernie was talking about earlier today. So multi-attribute method is really a way to use mass spec to characterize and analyze these biotherapeutics. And it really replaces a lot of different types of assays that you might have done before. And so this is the newest folder type in Panorama. It's something we've been working hard with some of our Panorama customers. And it really kind of gives you this quick pre-built reporting against that. So you can say, for this peptide, how much of a given sample had this oxidation on methionine at site 32 on this antibody chain. And so I can see, right, in this one, we've got 13, 14, and 18%. And I can compare that to both characterize which peptides or protein sequences are perhaps the most stable, as well as monitor the bioreactor output for some of the things like the question about quality by design and how do I monitor all of this going forward. So we're excited to keep building that out and definitely hope to have more connections between LabKey Biologics and Panorama, especially for these multi-attribute method type scenarios. I mentioned some of these features on the QC side, where these are premium features. So the majority of what you're seeing here in Panorama is part of the community edition of LabKey Server. But some of these kind of real customized usage scenarios are only for our Panorama partners, our premium customers, where you can configure exactly which metrics you want to be monitoring. You can add custom metrics as well. You can say, email me if I have 3 samples in a row that have at least 5 outliers, let's say. So you can kind of calibrate to say, right, 1 or 2 outliers, that's probably just noise. But once I hit a certain threshold, then I need to get actively involved and figure out what's going on. And then Panorama can also use Amazon S3 for file storage. So this is one of the areas in LabKey where we're able to directly import data from S3. We're looking to roll that out in more modules and use cases, but Panorama's kind of pre-configured at this point to go direct with S3 for storage if you've got a lot of data. So for next steps, encourage you to load up Panorama on your installation. We've got a lot of great resources, both in the LabKey documentation, as well as panoramaweb.org. This is the server I mentioned that McCost Lab runs, where they've got tutorials and lots of webinars about Skyline and Panorama. We've got also their Panorama Public, which is a repository for Skyline data and the raw files that meets the Proteome Exchange standards to support a publication. So when the journal says, hey, you have to share your primary data and your analysis results, this is a way to do that and then provide some additional context and using all the great collaboration tools like wikis and file shares that you've got in LabKey Server more generally. And then Panorama Partners is a variation of a LabKey Premium Edition subscription that gets you direct support from the Skyline and Panorama teams, from the McCost Lab and LabKey, including training and troubleshooting and bug fixes and all that good stuff that you're used to from your other premium editions. And it also gets you more direct input into our development plans. So if you've got, let's say new reports that you want to see in the multi-attribute method use case, let us know. And while we value the input from everyone, I won't lie, you get more of a vote if you're part of the Panorama Partners Program. And you get monthly calls with us and the McCost Lab team. And we've got a lot of great existing partners, pharma and biotech companies, as well as folks more on the academia side. So with that, can I call for joining us, take any questions? I think we've got a couple minutes here. Take a look at the chat window. Let's see a question on legacy MS data, mixture of targeted peptide and discovery runs. Is there much value in importing into Skyline and Panorama? So I think it really depends what you want to do with that. If it's really just kind of an archive that you're not planning to look at, I'd say don't bother, or maybe upload it just as files that you don't actually import into the database. But we have a number of groups that are doing a scenario very much like that. And if they don't already have that data in Skyline, what we've worked with them through the Panorama Partners Program to do is to have their new data come in via Skyline into Panorama, and then take basically an export of their legacy data in Excel or TSD or similar format, and then produce some reports that combine those together. So they have a unified report that they can see in LabKey, or they can see in external tools like what Rob talked about in some of the ODBC visualizations on his session at the end of yesterday, so that they can kind of mix and match, right? They don't need to reanalyze all that data. but they can have that unified reporting across both kind of the old and new. Any other questions before we wrap up for the day? All right. Well, I certainly would love to hear from you offline if you have interest in Panorama, whether you're using it now or thinking about exploring it. And also, if you see some of the ideas here that you want to carry over into other instrument types, other assay types, like I said, some of these QC workflows we think are really generally applicable. And we've got some other features outside Panorama to help you leverage the same general types of workflows. So before signing off, I want to remind everyone of the schedule for tomorrow, Thursday. So these are all Pacific time. So starting at 9 o'clock Pacific, we've got Hannah Brackey, who helped some of the questions on today's session. Who's our product manager for LabKey Sample Manager, and giving a preview of freezer management, which is kind of the biggest feature area that the team has been working hard to deliver in the past 6 or 9 months. I've got a great presentation from Patrick Layshot from OHSU on how they're using LabKey to manage their data for precision oncology. And then to kind of wrap things up for the day, we've got Steve Hansen from LabKey, who's going to show how you can load data into Sample Manager and then also see that on the LabKey server side. And we know that a lot of groups are interested in doing that, kind of maybe uploading assay data on one side and seeing it at the other, or vice versa. So some great tips for kind of how to use both systems in an integrated way. Thanks very much for joining us, and look forward to seeing you, at least in the participant list, on the call tomorrow. Thanks, everyone.